Welcome back for part 2 of our video series on biomolecules. In the first part we looked at carbohydrates and we looked at lipids, and in this part we're going to look at nucleic acids and proteins. So let's get started. What nucleic acids are. We can assume that they reside mostly in the nucleus, thus the name nucleic acids, but let's look at the functions and examples. Well, nucleic acids hold the genetic code. Talking about deoxyribonucleic acid and ribonucleic acid, or DNA and RNA. They also play a role in aiding in the synthesis of proteins. Specifically, the ribonucleic acid, or the RNA, will have a bigger job in that. Acids are made of repeating units of nucleotides. We would say that they are a polymer of nucleotides, and the nucleotide, the individual nucleotides, are the monomer units. So let's look at a single nucleotide. Now here are two generic drawings of nucleotides, slightly different, but each nucleotide has three parts: a phosphate group, a sugar, and some nitrogen base. Now the phosphate group is the same phosphate functional group that we learned about in our functional group video, but the sugar, let's look at the types of sugars this can be. There's two types of sugars that are possible here. One sugar is deoxyribose and the other one's ribose. Look at the structures and see if you can think about which one of these is ribose and which one's deoxyribose. So did you get it right? The deoxyribose has less oxygen in it. So these are the sugars, and notice again what we learned earlier on our part about the carbohydrates, that we end in OSE for sugar. So if the nucleotide is a DNA nucleotide, deoxyribonucleic acid, this will be the sugar. And if it's an RNA nucleotide, or ribonucleic acid, then the sugar will just be a ribose sugar. So what about this last part? Notice I drew it as a single ring here, but as a double ring on this side. These nitrogenous bases come in these two forms of single ring and double rings. We call them purines and pyrimidines. The purines are the double rings, and I remember that because they both have the letter U in them, and that makes the pyrimidines the single ring nitrogen bases. So if we look at the different types of nitrogenous bases, the purine double rings come in two versions, adenine and guanine, and the pyrimidines come in three versions: cytosine, thymine, and uracil. But uracil is only found in RNA. All the other four, three of the other four, are found in both. So that adenine, guanine, and cytosine are all found in DNA and RNA, but thymine is DNA only and uracil is RNA only. I always remember that the pyrimidines, cytosine and thymine, are pyrimidines because they have a Y in their name, and since uracil is going to take the place of thymine, then it is also a pyrimidine, and so that makes adenine and guanine the purines by default, and I remember purine by the U in double and pure. Here's a diagram showing the more specific the structure of the RNA bases, uracil instead of thymine here. Just a quick diagram that I found to show you. Now, if these are the building blocks, or the monomers from which we make nucleic acids, remember this is the brick, the building block. We need to build a much larger structure. We have to put a number of these monomers, a number of these nucleotides together to make a nucleic acid. So let's do that. I have a bunch of different nucleotides here, just generically drawn, and let's talk about how we'd put them together. It turns out that simply we have to connect the sugar of one to the phosphate of the next, and sugar to phosphate, and I can bring some more over here, and we could easily build these bonds. Now, I ask you if you think you know by what process this bond was made, and hopefully by now from watching the first video you know that that bond is built by dehydration synthesis, and we don't have to look at the details of that, and therefore they would be broken down by hydrolysis. So we could keep adding and build, as you can see, a polymer, or a long chain of nucleotides, which would equate to a nucleic acid. Now what I have here is a single chain of nucleic acid or nucleotides, and RNA is built in these single strands, but DNA is built in a double strand. And so to build DNA, we need to grab another chain, but we're going to flip them over. And when we do our DNA unit, we'll spend a lot more time on this. But if we take these guys over here, turn them around, and hold on one second. Let me do this. Whoops, come back over here. We could attach these guys, phosphate of one to the sugar of the next, building those bonds through dehydration and synthesis, and we'd have a double strand. Oops, this isn't going to work. Hold on, let me reorganize this, and I'll show you why in just a minute. The way I've drawn this can't work, because opposite every double-ringed or purine, I must have a single ring or pyrimidine. So better if I put them like this, and I would need, let's see, another one of these guys. And we'll put that down here, and then we'll connect them. And now I have two strands of nucleotides, and they would be held together by hydrogen bonds, these weak attractions. And so we could have a DNA molecule, which is a double strand of nucleotides. Like I said, we'll spend a lot more time on this in our unit on DNA, but I got the picture. We have some pictures of DNA molecule here, and again, we'll spend more time on this when we do the DNA unit. But there's our polymer of nucleotides as we're looking at the biomolecule of nucleic acids. Let's move on to proteins. Proteins are the structurally of the biomolecules the most diverse, and we should know by now that if they're structurally the most diverse, therefore they should also be, that's right, functionally the most diverse. So let's look at some examples of some functions of proteins. Well, structural. We build out of protein. For example, muscle fibers and connective tissue, collagen and keratin which makes the hair and feathers, the protein. So it's a building material. A transport material. We use proteins like hemoglobin's built from proteins. The membrane channels in the cell membrane and the pumps to transport molecules are made of proteins. Chemical signals. Hormones and neurotransmitters are made of proteins. Movement. For example, the contractile fibers in the muscles, the actin and myosin, are built from proteins. Immune response, antibodies, complement proteins, surface cell markers, proteins. And as catalyst, enzymes. We'll talk a lot about that in the next unit. So we see a lot of different examples of proteins, lots of different functions. We know that that means they must come in a lot of different forms or structures, because form relates to function. So we need to look at the function of proteins. So what are proteins made of? Proteins are made from individual amino acids or peptides. An amino acid or peptide, those are interchangeable terms. And the average protein is about 200 amino acids long. However, there are only 20 different amino acids found in humans. But how many different proteins can we make? That's like saying there's 26 letters in the English alphabet, but how many different words can we make? We know that putting those letters in different combinations and different numbers of letters, we can make basically infinite number of words. And the same is true for how we can build proteins. So let's look at the individual building block of proteins, the amino acid. Every amino acid is basically the same. You can draw amino acid as taking a central carbon. Carbon likes to make 4 bonds, and building off of that, on one side we just have a hydrogen, and then we need to look at what else we have here. Well, it's an amino acid. So let's think about our functional groups. We need an amine group, and it's an amino acid, so we need an acid group. So what group do we know, what functional group do we know is acidic? Well, how about a carboxyl group? The carboxylic acids. And the fourth part of every amino acid is what we're going to call the R group, and R stands for rest of the molecule. And it's the R group that can be different from one amino acid to the other. In fact, there are 20 different R groups. So there's your generic amino acid. If you can draw one, you can draw them all. But this is a monomer. Most proteins are many amino acids. In fact, we said around 200. So how do we put 2 amino acids together? Actually, before we get to that, let's look at this idea of the R group real quick. I pulled this diagram that shows all 20 different amino acids, and you can see in this that only the blue parts at the bottom, there's your R group. And you can see that there are 20 different versions of this R group, and sometimes it can be pretty simple, like this one here, which is just CH3 or a methyl group, and sometimes they can be branched and rings and just lots of different versions of R groups, but they're all unique. Here's charged, and so imagine that this makes the 20 different types. The other thing you'll notice in these drawings is that they have their carboxyl group drawn a little different, and they have their amine group drawn a little different. So let's address that real quick. You may recall from our functional group video that carboxyl groups are acidic, but also amine groups are basic. So we know that acids like to give away hydrogen, or H plus, a proton, and bases like to pick those protons up. So this hydrogen here, or actually a proton, we'll make it a proton, will be donated by the carboxyl group and often end up over here on the amine side because bases like to pick up protons. So they've already just drawn that in these pictures with this H already on this side, which is what will really happen. So now let's put some amino acids together and build a protein. So here I have two amino acids with the carboxyl end of one near the amine end of the other. And if we're going to put these two together, let's think about what we can do. Well, if I took this OH off and this H, what does that make? There we go. So we make water. So we pulled water out, or dehydrated. And what do we have left behind? Well, we have a nitrogen that needs another bond, and we need a carbon that needs another bond. So we could put those together and synthesize a bond. So dehydration synthesis. Now this bond has a special name. It's the bond between two amino acids, and another name for amino acid is peptide. So this is a peptide bond. So now I have a dipeptide, and you can see that if I grab another one and put that here, we could do the same thing again, pulling out water and building another peptide bond, and we could quickly make a tripeptide through dehydration synthesis, making another peptide bond. Again, we're making a polymer. If we look at this polypeptide, many peptides, many amino acids, we can see by counting R groups how many different amino acids we have here. And we could draw arrows to peptide bonds there, there. Oops, I lost that one there. And make sure you get the right bond if you're marking peptide bonds. It's the bond between where one carboxyl group used to be and where one amine group used to be, so it's right in between there are your peptide bonds. Now, the question is, how would you take this apart? If we built it by dehydration synthesis, pulling out water to build a bond, we would then digest it or take it apart by adding water back and breaking that bond with a process of hydrolysis. Hydro, water, lysis, meaning to cut. And in this case, how many water molecules would we have to add back in? Well, one for every peptide bond. So if I have 1, 2, 3, 4, 5, 6, 7, 8 amino acids together, it would require 7 water molecules to break those 7 peptide bonds between them. So that brings us to our concept about the structure or the shape of proteins, because as we draw this, it looks very linear. We can see it's like a straight line, but in fact proteins are not in straight lines, but that is their primary structure. So we can think about the levels of structures of proteins on 4 different levels. The primary level is the sequence of amino acids. It's the order we put those the letters of like if we're spelling out a word, the order we put the letters in is the spelling of the word, and it's a linear idea. But proteins don't stay linear. In fact, as we look on the backbone of this molecule, let me scroll down here, there will be a lot of hydrogen bond interactions in this backbone itself, in this linear part, that will cause the molecule to twist and kink in a repeating pattern of coils and folds due to this hydrogen bond. So that gives us a second level of structure. And then if we have, so think about like a slinky or something. And then if you took the slinky, which is already coiled, and folded the coils on itself, you get a third level of structure. And this third level of structure, these irregular contortions, is due to bonding interactions between R groups. Imagine if each of these R's hanging off is, remember, it's not just a one thing. That's a could be very large parts hanging down there. So you have something coming down off of here and something come off here. It could be branched, and you could have rings and all different things. And some of these are going to attract to each other. Some of these are going to be repelled from each other, and that's going to cause this molecule to kink and fold and twist on this third level of structure that we see here. And then on a fourth level of structure, let me scroll down, or our quaternary structure, is when we have more than one polypeptide chain. So more than one polypeptide chain. Now this structure is very important because we know that form relates to function, and when we do our protein synthesis unit and our DNA unit, we'll talk a lot more about this, how the structure of the protein is formed and how that form relates to its function. It'll be a big concept when we discuss enzymes also. But in talking about that, we'll briefly introduce the idea of denaturing, or the losing of shape. If a protein loses its shape, it's usually due to the disruption of R group interactions and the hydrogen bonds due to the effects of changes in pH, temperature, and salinity, which causes this protein to maybe uncoil and untwist and unfold, and when it loses its shape, it loses its function. So now we're at the end of our video, or two videos on biomolecules. To review, you might build some kind of chart like this where you look at functions, examples, building blocks to other information for the big four. Also, you might want to go through and see if you could by sight name these molecules that we see, and you have them in your note packet, so just look over that and email me if you have any questions.